Yep.
So, welcome everyone. Thank you so much for joining us for today's special edition FSHD University webinar on the Fulcrum Phase 3 clinical trial. We are very excited to have Dr. Jeff Statland here with us today to give a presentation on what the outcome of the Fulcrum Phase 3 trial, the implications to future research, and so forth. A quick reminder. Everyone, if you can please make sure to use the Q&A session for any questions that you have. During our question and answer session, we will answer those that are received that way. The chat will not be monitored, so please do that. And then a reminder that this session will be recorded and available on our YouTube channel. And with that, I would like to introduce Dr. Statland. He is a professor of neurology at the University of Kansas Medical Center in Kansas City. And his specific research interest over the last 10 years has been preparing for clinical trials in FSHD. He has worked with collaborators to develop new disease-relevant outcome measures to assess patient-reported disease burden, functional impairment, and physiological changes in muscle. And he's also the co-principal investigator for the FSHD Clinical Trial Research Network, which we refer to as a CTRN. And it is comprised of 32 institutions in the United States, Canada, the United Kingdom, European Union, Brazil, and Australia. Dr. Statland frequently volunteers his time with us, and we are so grateful to have him here to present with you all. So with that, I will hand it over to Dr. Statland.
Okay, fantastic. And let me just pull this up. Are you able to see the slides?
Yes, you are good. Thank you,
Okay,
Dr. Statland.
great. So I want to thank the FSHD Society, and I also want to thank Fulcrum Therapeutics for allowing me to share this top-line efficacy and safety data from their phase 3 placebo-controlled trial of Losmapimod for FSHD. Now, while it's disappointing we didn't see a clinical benefit in this study, I do want to take a moment to reflect. This was really our first phase 3 disease-targeted trial for FSHD that was really ever conducted. And it paved the way for the subsequent companies we're seeing moving into the space now to pursue their disease-targeted therapies. And it also allowed us to validate outcome measures that are going to be important as we move forward in the clinical trial programs, including things like whole body MRI and reachable workspace, which were really developed as part of the Fulcrum program. And it really showed other companies that there's a regulatory pathway to run these phase 3 studies. So I think important for the space to really set the stage for clinical trials as we move forward. So as everyone knows, FSHD, it's one of our most common muscular dystrophies, about 20 to 30,000 people affected in the United States. There's two genetically distinct types of FSHD, type 1 and 2, but they converge on a common downstream pathway. So in both of them, you get de-repression of the transcription factor DUX4, which causes the disease by a toxic gain of function. And so what this means for us is that if a drug targets DUX4 or any of the downstream pathways of DUX4, they can include both FSHD1 and FSHD type 2 and probably be effective for them. Clinically, both types present in a similar fashion. There can be a lot of variability between individuals and from even one side of the body to the other. But overall, we have a chronic and progressive skeletal muscle condition that leads to loss of muscle mass, muscle strength, and muscle function. About 20% of people will lose the ability to walk or to maintain a job because of FSHD. We have no approved treatments currently available. So our treatment is multidisciplinary clinics, it's supportive care. So there's a critical need for effective therapies for FSHD. Losmapimod is a repurposed, orally administered small molecule. It's a selective p38 inhibitor. It was identified from patient-derived cell assays based on its ability to reduce the expression of DUX4. It had previously been tested, though, in clinical trials for diseases like COPD and rheumatological conditions. So more than 3,000 patients had been in clinical trials where they had exposure to Losmapimod, and overall, it had appeared to be very safe for those individuals, if not effective. There was a phase 2 study that Fulcrum conducted called REDUX4, and some of you may have participated in that. In that study, unfortunately, they were unable to show that Losmapimod affected a whole series of downstream DUX4 targets in muscle biopsies. But that said, they were still able to see a clinical benefit in strength, in function, as well as muscle quality on MRI and patients' report of improvement. And because of that, we felt like there was a need to run this phase 3 study. And so for this study, it's a global randomized placebo-controlled parallel group multi-center phase 3 study, which is a mouthful. The eligibility criteria. So you had to have a genetic diagnosis of FSHD, either type 1 or type 2, verified by genetic testing, clinical severity score of 2 to 4 on a scale that ranges from 0, which would be unaffected, to 5, which would be requiring a wheelchair. 2 to 4 would be individuals who have some shoulder weakness and may have some difficulty getting around with lower extremity weakness, but still able to walk on their own. They had to have a reachable workspace area, and we'll return to this in a second, but that value had to be between 0.2 and 0.7. And what this means is it's in the dynamic range for what we measure in reachable workspace. So weakness in the shoulders, but still able to reach up at least a little bit. The primary endpoint for the study, so this is the outcome that will determine whether the drug works or it doesn't work for this study, is a change from baseline in the average reachable workspace area with a weight attached from baseline to week 48. And this is the average of both arms. And we'll return to this just in another slide where I'll show you what this is. Secondary endpoints were really a whole range of endpoints that included patient reports of function, muscle quality on MRI, and strength testing, including things like a global impression of change, the change from baseline in a whole body composite of muscle fat infiltration in what are called B muscles, and we'll discuss this a little more. A relative change in baseline for the strength of shoulder abduction, a change from baseline in the neural qual upper extremity scale, and then measures of safety and tolerability. So when we look at the study, 354 individuals were screened for the study. 94 of them didn't meet the criteria. And when we look at the reason why people weren't meeting the criteria, the most common reasons were related to that reachable workspace area or that severity score at baseline. 260 individuals were randomized, 130 equally to each group, Losmapimod and people on placebo, and 126 in each group completed the study. When we look at the readout for the study, the primary outcome is the reachable workspace. This is a novel repurposing of an existing technology. And some of you may remember the Xbox, the game console that came with the 3D camera. This is that same 3D camera that they repurposed to develop an outcome that assesses the region of space people can reach into. So a standard set of movements that people do, and it creates a volume of space, your reachable workspace. You can see it illustrated in the bottom right of the screen. There's 4 quadrants that surround the individual, and then a 5th quadrant, which would be reaching back in behind the individual. When we look at the values of the reachable workspace area, it does produce a number, which on its own may be a little difficult to interpret. But if you look at the scale of that number, how large or how small it is, it does correspond to the ability to do activities. So people doing things like putting an object up onto a shelf or taking a shirt on and off, they'll have higher reachable workspace area, where people who have difficulty with those tasks would have a lower reachable workspace area. Muscle quality was also evaluated in this clinical trial, and it used this novel whole body MRI technique, which uses AI to help segment the muscles and then do the measurements of 38 different individual muscles that go into the data that's used in this clinical trial. They were particularly interested in what were called B muscles. So sort of like that criteria around the reachable workspace area, you can think of B muscles as muscles that have some involvement on MRI, but are not completely wasted in appearance. So fat fractions between 10 and 50% at baseline. These muscles that are actively undergoing change are the most likely to change over shorter time frames, and this had been shown in prior studies. What they actually looked at was the muscle fat fraction, which is the average fat across the muscle. The fat infiltration, this is just the fat fraction, but only in the parts of the muscle that have less than 50% fats or are thought to be more susceptible to changing. And then the lean muscle volume. This is the volume of the muscle where the percent of that that's thought to be due to fat is subtracted out. So overall, the participants in the Losmapimod and the placebo group were relatively equal in terms of age, in terms of the assignment of the gender, in terms of body mass index, so weight, and even in terms of the frequency of people who had type 1 or type 2 FSHD. And that frequency we see about 93% with type 1. This is typical for what we see in a general population. There were some criteria that the company was interested in looking at because there was at least a suggestion that they might be associated with different trajectories over time. One of them was in people with FSHD type 1, they would look at the number of repeat units that were remaining, in particular looking at that 1 to 3 repeat units, because these individuals are supposed to be getting symptomatic at earlier age and maybe having a more aggressive disease course. You can see a slightly higher percentage of individuals in the Losmapimod group with 1 to 3 repeats compared to the placebo group, and that's 16% compared to 11%. The other thing they looked at was the severity score at baseline. In particular, they were looking at those individuals who were more severely affected at the first visit. And you can see that there's a slightly higher percentage in the placebo group at 48% compared to 37% in the Losmapimod group. And so a little bit conflicting in these two groups. More individuals with 1 to 3 repeats in the Losmapimod group, but slightly more individuals who were more severely affected at baseline in the placebo, perhaps counteracting each other. From this slide through the rest of the results slides, they're all going to be oriented the same way. On the left, you're going to see a graph that gives you the week-by-week changes on the outcome for the population. 0 is going to be no change. In this case, we're looking at the reachable workspace area. So going up would be getting better, going down would be getting worse. In all of the graphs from here on out, placebo will be in blue. Losmapimod treated patients will be in orange. On the right, you see what the actual statistical test will be done on, which is that change from baseline to 48 weeks. And you can see the change on the y-axis, and then again, placebo in blue, the treated in orange. So what you see here is that both the placebo-treated patients and the Losmapimod-treated patients improved in their reachable workspace area over the course of the study to essentially an equal amount. This is different than what was seen previously in the phase 2 program, where the patients who were in the placebo group lost function by about a similar amount to what you see here. So in this case, the primary endpoint, which is what determines whether the study is positive or negative, wasn't met. There was no benefit in the reachable workspace area. This is looking at the MRI muscle quality. This is looking at the muscle fat infiltration averaged over an index of those B muscles. These are the muscles that are actively in the process of changing. You can see that both groups, in this case, the placebo and the Losmapimod patients, increased in the fat infiltration over the course of this study by about less than slightly 1%. So not a large change, but consistent. You can see that that change, though, was less for the Losmapimod-treated patients than it was for the placebo-treated patients. You can see that difference then illustrated on the right. This also doesn't distinguish itself out of the normal variability of what we see. So not a significant effect. Compared to the phase 2 study, the placebo group changed to a similar degree than what was seen in that phase 2 program. The difference here is the Losmapimod treated patients in the phase 2 program showed almost no change, where they're showing an increase in the fat infiltration here. Looking at strength, and this is looking at the strength in the shoulder abductor muscle as measured by handheld dynamometry. Again, like with the reachable workspace, you can see that both the placebo and Losmapimod treated patients are improving in strength over the course of the study. And you can see that the size of that effect is not too bad, about a 10% increase for Losmapimod and just around a 2.5% for the placebo, which might look like a benefit for the Losmapimod treated patients, but the variability in this estimate is quite high, and so it's not able to distinguish itself out of just the noise of the measurement. So no effect seen in strength. Comparing this to the REDUX4 study and looking at the average of what they saw over the shoulder abductors, in that case, the placebo group, like for the reachable workspace, had decreased in strength by about a comparable amount over the course of that study. Looking at the patient global impression of what happened over the course of the study, this is a questionnaire where 4 would be no change from previous visit, 3 would be a slight improvement, then 2 would be a large improvement from the prior visit, going up on the scale, so to 5 or 6 would be getting worse. Over the course of the study, you can see that both groups actually state that they're improving over the first half of the study, but you can see by the end of the study, patients are saying they've really perceived no change, and this is the same whether you're looking at placebo or Losmapimod treated patients. And then the last patient-reported outcome is the NeuroQoL upper extremity scale. For this scale, going down on the values here would be getting worse or losing upper extremity function. You can see that both the placebo and the Losmapimod treated patients are losing some function in the upper extremity on this scale, with the Losmapimod treated patients losing slightly more function, but this also doesn't reach statistical significance. When we look at safety and tolerability, overall, Losmapimod was really well tolerated. There were no serious Losmapimod-related adverse events. When we look at the most frequent adverse events that occurred in the study, they're similar to what was already known for Losmapimod, and most of them were grade 1 or 2, which means they were mild to moderate in severity, and all of them occurred at a frequency less than 5%. So headaches, diarrhea, nausea, GI upset, and rash or dry skin. There was one serious adverse event that I believe was appendicitis in the placebo group that was thought to be related to treatment by the investigator. And there was one death in the study. It was thought to be unrelated. This was someone who developed lung cancer over the course of the study and unfortunately died. So overall, unfortunately, Losmapimod didn't show a clinical benefit when looking at the reachable workspace over 48 weeks, and also didn't show clinical benefit when looking at muscle quality on MRI, when looking at strength of the shoulder abductors, and when looking at patient reports of change. Participants in both the Losmapimod and the placebo groups actually improved in the reachable workspace area over the study, which was in contrast to what was seen in that prior phase 2 study, where the reachable workspace declined in the placebo group. There are several reasons why this may have occurred. The first would just be that that first study wasn't really representative of what we see in a general population. And so this study really showed us what we would see in an untreated group. Other possibilities would include that the duration of the study was just not long enough to see those clinical benefits. So a longer study may have allowed those groups to separate out. It's possible that enrolled participants were aware of the prior phase 2 results, and so they came into a study with an expectation that they would get a benefit, and that may have affected performance on these outcome measures. And there were some differences in the eligibility criteria when we look at their phase 2 study and this phase 3 study. The phase 2 study did not include people who have FSHD type 2, although I think that's unlikely to explain these differences, as the numbers were equally assigned to placebo and treatment here, and the phase 2, the type 2 population did about the same as the type 1. In the phase 3 study, they did require people to have a certain amount of weakness on shoulder abduction, but still be able to do that task. This hadn't been required in the phase 2 study. It's unclear whether this would be enough to explain the differences that we're seeing in these studies. And then finally, in the phase 2 program, there was an MRI entry criteria that required people to have STIR-positive muscles on their MRI. These are thought to be muscles that demonstrate active molecular changes in the disease and thus are more likely to change over a short period of time, and thus may have selected a group of people who were more likely to show some progression. Overall, the drug was safe and well tolerated, but unfortunately didn't meet its goal, so didn't show the clinical benefit. That said, I think a lot of pluses that came out of the study, including an example of a regulatory pathway for companies coming next, showing that we can get FDA-approved protocols through phase 1 to phase 3 that they would accept for these studies, and then validation of new endpoints that are likely to be important in the space. So I really want to thank everyone who's attending here and everyone who participated in the study. It was the participation people like you that made a large international effort like this actually happen, and then all the people who took part in the study at the different sites. It takes large teams to do studies like this with investigators all over the world. There's study teams and multiple companies helping to run a study like this. All right, I'm happy to take some questions.
Great. Thank you so much, Dr. Statland. Yes, we do have questions coming in. I think one here is, do you have any thoughts as to why the phase 2 and phase 3 outcomes were so different?
Yeah. So I mean, I think they're referring probably to the reachable workspace. Yeah. So, it's hard to know. When we look at our natural history study that some of you may have participated in, what we see in the sort of average adult who enters that study is that there's really no change in the reachable workspace over a year. Um, so it's a little hard to know. A slight improvement might be expected in an interventional trial. In prior trials in FSHD, looking at things like strength, we do tend to see people improve in their performance once they get in a drug trial because everyone's very excited to be in the trial. We often repeat those measurements multiple times during the conduct of the trial so people learn how to do them more. Now, the initial phase 2 study that showed that more rapid decline in the shoulder range, the reachable workspace, maybe was due to that MRI criteria, but it's hard to know because there is an effect that you see in trials as you go to these larger populations where the size of your effects start to get smaller and smaller. This is called regressing to the mean. It just tends to account for the greater variability we see when we include much larger and more representative groups of patients.
This next question maybe gets to whether could there have been subsets of patients, such as people have, say, smaller numbers of repeats, or lower Ricci scores, or anyway, some kind of subset where you might have expected to see a larger benefit from Losmapimod.
Yeah, I do love that question. There are a lot of people who would love to have access to this data set and do that analysis. But you do need to remember a couple things. Statistically, pre-planned analysis will have some power. So you can be fairly confident that statistically, if you see those differences, they're real. A classic mistake people make with a large study like this is you start looking at lots of subgroups that really weren't planned in the analysis. You decide you see an effect, you repeat the study with a larger group, and then you discover there's really no effect there because you've selected for the difference. There are some pre-planned analysis, though, here. That included the individuals with 1 to 3 repeats and the individuals with increased severity. For their primary outcome, they didn't see any differences. For the MRI, however, maybe there's a trend towards a benefit for the drug in the people with the 1 to 3 repeats. And that data should be released soon with the publication of the trial results.
Okay. Here's a question that maybe gets a little into the molecular mechanism. I said I'm not quite sure I'm interpreting this correctly, but based on knowledge of potency and plasma protein binding, do you have a prediction for the level of p38 phosphorylation and the expected DUX4 reduction based on the exposures achieved in the human?
It sounds like someone who has some experience
Yes,
with
some
drug
scientists out there.
development. I mean, I only know is what the company suggested, which is they felt like their dose was maxing out the effect at the enzyme for the p38 inhibition. But this is not based on something that's DUX4 related, right? This is just looking at the general, I think, enzyme activity And this would fall into the category of data that may or may not get published. I don't know what the company is planning to do with that.
Here are a couple of questions related to some of the clinical endpoints, whether there are other endpoints that should be considered, such as timed up and go or pulmonary function tests that might apply to a broader or different kind of demographic. Someone else is asking also about functional measures addressing lower extremity strength. And then, well, let's stop there for a minute. There'll be.
Yeah, so if you had come to me cold 10 years ago and asked me what outcomes do I think we should include in a clinical trial, I likely would have said these functional measures that are assessing mobility, mainly because, you know, in the average adult, often there's shoulder weakness there that's sort of happened for a lot of people. So not actively changing a whole lot, whereas the muscles that help us get around do tend to be in that active phase of changing. This is what we see in the natural history studies. Those outcomes tend to be a little more sensitive. It's always hard to know in a trial, though, which is going to be the best outcome for what the drug does. Now, Fulcrum did include other outcomes in their phase 2 program. So they selected the ones that were in phase 3 because these were the outcomes that had the largest effect in phase 2. That may have been a mistake. They did include the timed up and go in phase 2. It showed a trend towards a benefit, but it didn't statistically separate out within the phase 2 study. They also included other muscles, including muscles in the pelvis or legs that also showed some benefit, but statistically didn't differentiate itself out because the numbers were somewhat small in the phase 2 program. But the company selected to go with what was in the phase 3 program.
And this is somewhat of a little bit hypothetical, since we don't really know yet about how robust reachable workspace will prove to be. But they're asking about in a non-ambulatory group, one could imagine there would be progression that you could be measured with reachable workspace. Could you envision that the FDA might approve a drug based on reachable workspace in a non-ambulatory population? And
Yeah,
again,
I mean, I
the
think a
natural
few
history.
things to say about that. One, the FDA was obviously open to the reachable workspace. They allowed it to be the primary readout for a phase 3 study. I think they probably wanted to see some consistency across the other measures in this case. I think the reachable workspace would be a very good outcome for people who, can't walk anymore. You may need to be adjusted slightly for those individuals because of some changes in their ability to sit in the chair while they're doing the task. But I do think, me as someone working in the space and developing tools, we're very centered on this idea of having outcomes for everyone with FSHD, whether they're walking or not walking, wherever they're at, whether they're children or adults. And so we're trying to get an array of outcomes that people could use in all of these scenarios, because we don't want that to be a barrier to drug development. We're trying to make sure there's options for whatever they think the treatment might do.
Okay, right. There's a question, is there any thought to a follow-up, say 6 to 12-month study after the discontinuation of the drug? This individual says she was a participant and feels, I have significantly declined after being on the drug and this has been validated at my annual neurology appointment. So yes, I guess a natural history on people who were in the trial and have stopped. Is that something being looked at?
Yeah, I mean, this is it's unfortunate. So this tends to happen in every study. When you look at the population, there's often people who appeared to respond to treatments and then people who didn't. On average, we saw no benefit, but that doesn't mean it didn't benefit, that individual. Unfortunately, right now, I mean, the company themselves don't have a plan to move forward with development of this drug. Someone else might, and this will depend on factors like the ability to get access to the drug, which I think is currently in the hands of Fulcrum. But I do think there would probably be interest in trying to understand, was there a group who might have benefited? It's a small molecule, it's an oral drug, it has a really nice safety profile, right? People were on it for a long time, 6 years. I think if we could find a group who would benefit from it, we would love to, have this as an option. But it probably remains to be seen right now whether we'll actually see that happen. But we might see it with other molecules that would sort of be not that dissimilar from Losmapimod, that might be a pill, something that would be easy to take with a fairly good safety profile that might have a disease-targeted effect on FSHD.
I've just mentioned the society is in the process of having that clinical trial data transferred to the society. So we are to be able to start doing that analysis and seeing if there are subgroups and seeing if there's a possibility to design a more, a different kind of trial. And certainly there are advantages to working with Losmapimod since it's already been thoroughly safety tested and there's a lot of knowledge about it. So that's one possible direction that we in. And as you said, Dr. Statland, there are a lot of benefits to having an oral pill with a good safety profile in terms of people being able to access it. And if it's a fairly moderate benefit, if there's any benefit at all, that's in FSHD right now, right? So let's see, this is more of this is a comment, I guess, from an individual. Hello, I was previously enrolled in the REACH study with Losmapimod at WashU, Washington University, and he strongly disagrees with having the cessation of the phase 3 study, as I believe we are missing the forest for the trees. The measurement endpoints that were focused on did not adequately capture the benefits that I and others I have spoken with saw. I experienced a reduction in falls from twice per week to around every 6 weeks. I do not believe you can glean appropriate data to make a conclusion based on the reachable workspace and MRI. The measurements that Dr. Pestronk had taken showed functional strength improvements in a wide range of motions and activities. Why were these not considered? I have had steady decline for 25 years straight, and then suddenly my first uptick in measurements only to have the study medication taken away from me. Totally unfair. Even in the absence of dramatic improvement, has anyone stopped to think not getting worse is a victory for us patients?
Well, I mean, I think the study was powered to see a halt of progression. So, I mean, that was probably what they were hoping for. It's probably worth pointing out their phase 2 program had additional outcomes in it. It included, while not exhaustive, it included multiple muscles besides the shoulders that they measured: the elbow flexion, the ability to straighten the elbow, the knees, the ability to sort of extend them or bend the knees, and the ankles were all measured. They opted to go with what they went with because they chose what were the largest effects for their phase 2 study. We could argue about that decision, but that's a fairly typical decision that someone makes as they go from their phase 2 to their phase 3 program. But I do agree with you. This is always a problem with any drug development, is what do we do when people are benefiting, right? And on average, the drug's not showing that benefit. And that's a difficult, it's a difficult challenge. In an ideal world, we could still develop studies where we would make that available, in a personalized way to the individuals who might be benefiting. I mean, that's something that I think we can work towards in the future.
Is there a path to doing that with a drug like this through some kind of compassionate use?
Yeah, if the drug was made available, you could do these what are called N of 1 studies, or studies in single individuals. It's a little bit challenging because there's a workload at the site to do it. So typically, if a company is going to make it broadly available to individuals, they call it an expanded access program. And then they'll organize it in a way where they can offset the cost to do it. So it's logistically a little bit challenging, but it could be done, and there is a regulatory pathway to do that. That's true.
But it seems unlikely Fulcrum itself would.
Yeah, they're not likely
It would
to pay
yeah.
for that at this point, because
It would need
they're
funding
not.
from another. It would need funding from another source. But
Yeah.
you see there is a pathway to doing that.
Yeah, so if we could get access to the drug and there's interest, we could probably set up a program like that, but it would need to be funded.
Yeah. Okay. Question here is, did participants find out whether they were on drug or placebo at the end of the trial?
Participants should find out if they're on drug or placebo. I need to go back and just find out if they released that information. I think one challenge for a company like Fulcrum, which came together for this drug, is the company also, dissolved largely at the end of that trial. And so, but this is something that typically is released, and there's no reason at this point why it shouldn't be released.
Would it be the company that releases it or the site?
Yeah, the company has to release it.
There's a question, do you think it would be better to go after DUX4 more directly rather than indirectly, as in the case of going through the P38 pathway?
That's an easy question for me. If you're asking my opinion, yes. And I'm very excited about our molecularly designed, gene-targeted therapies. I'm quite enthusiastic about the possibility of getting larger clinical benefits with those types of drugs.
There's a question here really for the society. It says, is the society actively looking into compassionate use funding? And is that something that the society or another organization like the MDA would back? I would say right now that we're looking at understanding what is involved, who would need to be involved, what kinds of clearances, access to drug is required, what that would cost. So it's a little hard. There's not like a clean checklist or something somebody hands to say, this is what it would cost to do a compassionate use study, but and it's significant dollars. I don't know, Jeff, do you have a ballpark idea of what something like that would cost
Well,
to set up?
it's a little challenging. The way the programs are set up with the FDA, and each country will regulate this slightly differently. They do have pathways for compassionate use. It's usually in the setting of a drug development program. So they want someone to be doing the regulatory requirements to show the drug safe and effective, and then they provide access to more individuals. This helps us with safety and understanding how people who aren't in the trials respond. It's a little harder to know what this would look like if there was no one pursuing that development, right? We're just going to take molecules and give people access, because the program's not really meant to be an in-run, around the ability to make sure we have safe and effective drugs that are out there. That said, we could talk about it. The costs are largely costs for those sites to do the regulatory filings that would be required and to collect any of the assessments that need to be collected. Typically, in a compassionate use program, you bill insurance, and your insurance may or may not cover those costs like anything else. Every policy will be a little different. And so that could be an out-of-pocket expense for people.
Mm-hmm. Unless we're able to find
Unless someone funds it, that's
sponsor,
correct.
right?
Yeah.
but the company isn't there to sponsor
Yeah,
it.
this is why in the larger expanded access programs, typically they're not administered by each local site, right? They're administered by a company who does it centrally.
Right. There was a question, has anyone looked at the data to look at whether the individuals were participating in regular exercise routines or not? Is that
Yeah, that's a question after my heart. I think that's a fantastic thing to look at. No, it would be the easy answer to that. I mean, there probably is data to look at, and hopefully when the data sets are in a place where people can query them, we can see if they collected enough to answer that question. But I do think that's an important question and an important concept as we think about our trials and our drugs. How would they work in the setting of exercise or adding activities?
Mhm. Right? There's a question if people who participated in the Fulcrum study, either the REACH or the Redux 4, I guess, would they receive any priority in enrolling in future clinical trials?
Yeah, I mean that's a hard question. The truth is, is Fulcrum's not, pursuing their program. So not, I think, priority in the way that a company might prioritize someone who previously been in their own program. They'll often, for people who are in their early phase studies, right, that are short and right, don't really give you a lot of access to the drug, they may create what's called an open label study where they can then give people access to the drug at a later time point. Um, the each site kind of has their own process for prioritizing for trials. What I'd say is reach out to wherever your care is and make sure they know you want to be in clinical trials. And most sites try to do this equitably. So, first come, first serve for people who are interested and qualify, and then for people who've reached out already, get some priority that way.
I think if in addition to
Yeah, I apologize. I'm not sure I'm hearing.
in files, I guess maybe it seems to have frozen.
June, you're breaking up. Can you repeat the question again? We'll see if it comes through this time.
Can you hear me?
Okay, we can hear you now.
I'm frozen.
There we go.
I'm the question whether I think getting it the header, some have lower extremity involvement.
June, you keep breaking up. I
Maybe
can
you
go
can
ahead and
turn the
ask this
video
question.
off and see if it works better.
Oh, okay. Okay. Can you hear me now?
Okay, Dr. Statland, I'll kind of step in right now and ask
Yeah.
the question. Yes. So the ACE 083 trial also had a similar system for imaging, walking, lower extremity mobility. Possible a future trial using both imaging techniques to analyze both the upper extremities, RWS, and the lowers for a more complete overall assessment. So is it possible for a future trial using both imaging techniques?
Yes, of course it's possible. And in fact, it's being done. So the current studies are using a whole body imaging technique. It has about 138 muscles across the body from out at your feet all the way up to your head, that will be imaged. And they'll take composites across the whole body. And then certainly what the other things we measure or functional measures would make sense to have something around mobility and something around the shoulders and arms.
Thank you. Another question that we have. So, can this study be repeated with a different set of people to see if it's consistent?
Yes, I mean, you can always do that, and indeed, it has been done. I think in diseases that are more prevalent, for example, diabetes or congestive heart failure, where there's a lot more people affected, it is very common to do at least 2 phase 3 studies, and it's partially for just what you're saying, to get a better sense of the generalizability, and because sometimes you get results that are what I would call indeterminate, right? We're not sure whether it's really working, but we're not sure it's not working. And having that other study could help answer that. It's very hard to do that in a rare disease where running the study is maybe more expensive, or the companies are much smaller, right? They don't have the finances to do that. So the model for rare diseases has often been to just do 1 phase 3 study, but it's certainly possible to do another one. And for more common diseases, that would be considered a standard to do that.
Beautiful, thank you. Another question that we have is, how did the results from the placebo group in the phase 3 REACH study compare to natural history results regarding clinical endpoints?
Yeah, that's a very good question. I think for the reachable workspace itself, probably the natural history does just slightly worse. So probably over 48 weeks you would see really no change, whereas the placebo group in this study maybe increased by 1 or 2%. So a very small benefit. I doubt that those groups would separate out. If we just compared the placebo here to the natural history, they would be very close. For other outcomes, it's a little harder to know. We don't have a whole body MRI and a large natural history data set yet. We're collecting it now. Any of you who are participating in the MOVE Plus natural history study, the plus is the whole body MRI, and we're collecting it for just this reason.
Great. May have
Let-
gone
Let's
through all
see.
questions.
Okay.
I think that's all our questions. Yes, and I'm sorry, my computer was starting to freeze up too. Zoom is being finicky today. Thank you so much, Dr. Statland, for
Yeah,
taking
my
the time to be here
pleasure.
with us. Yes, no, we appreciate it. And thank you, everyone, for joining us today, despite the Zoom tech issues. We made it through. And thank you for all of your questions. We really appreciate you, Dr. Statland, as always. I encourage everyone to please look at our events calendar, as we do have a number of events coming up within the next few weeks, especially with World FSHD Day around the corner. We'll have a number of celebrations. We also have a number of in-person and virtual chapter meetings, as well as future webinars. We have another FSHD University webinar coming up on, let me get the date for you. It will be on Thursday, June 26th at 5 p.m. Please note that this webinar is not at our usual time due to Dr. Bennett's clinic schedule, but we look forward to seeing you. Again, thank you, everyone, and we will see you next time. Have a wonderful day. Thank you, Dr. Statland.
Thank you.
Thank you, June.
Bye.